Welcome all to the next class of AK Bio lecture series. Today's lecture focusing on the branch of genetics, the cytogenetics. As zoologists, we all heard the term cytogenetics in different occasions, and in modern era of clinical genetics, the experts depend different cytogenetic techniques as experimentally and clinically as part of their profession. Before starting the discussion on the technique in detail, we have to travel back to the history of cytogenetics. It was one Swiss botanist named Nageli who first described a thread-like structure in the nuclei of plant cells in the 1840s and what he called transitory cytoblast. It is now known as chromosomes. Later in 1888, Walder, another scientist, coined the term chromosome after staining techniques had been developed to make them more visible. Actually, chromos, the term chromos, is a Greek word for color, and the term soma is a Greek word for the body. The field of human cytogenetics was initiated in 1956 when the number of chromosomes in a diploid human cell was accurately determined to be 46. Cytogenetics is a branch of pathology and genetics concerned with the study of chromosomal structure, location, and function in detail. It includes the study of chromosome number and appearance, that means karyotyping, the physical location of genes on the chromosome, and chromosomal behavior in processes such as the cell division. And cytogenetics also includes the study of factors that cause chromosomal changes. And chromosomal aberrations can be either constitutional, means present from birth, or acquired. As the field of human cytogenetics emerged, researchers began to develop the methods to visualize chromosome structure and organization. Scientists quickly realized that not all chromosomes are created equal specifically, they differ in their length and in the position of their centromere. And researchers also focused on numerous studies to determine the relationship between the human diseases and chromosomes. As part of early cytogenetic studies, the studies revealed that an extra or missing copy of certain human chromosomes could lead to diseases. For example, in 1959, an extra copy of chromosome 21 was shown to be associated with a Down syndrome. In the same year, several abnormalities in sex chromosome number were linked to Turner syndrome and Klinefelter syndrome. Soon after discovering the link between chromosome number and disease, researchers turned their attention to the role of chromosome structure. Thus, in 1960, uncovered a link between chronic myelogenous leukemia, that is CML, and abnormal chromosome structure. The researchers discovered the presence of a small chromosome which they named the Philadelphia chromosome, that was unique to CML cells. Then in 1985, scientists studied the chromosome of chronic myelogenous leukemia patients and found that in majority of patients there is a fusion of parts of BCR gene from the chromosome 22 and ABL1 gene from chromosome 9, resulting in a gene fusion product called BCR-ABL protein fusion protein. The branch of cytogenetics can further subcategorized into two. First one, classical cytogenetics, and second one is molecular cytogenetics. The classical cytogenetics allows microscopical visualization of a whole chromosome to access their number and structure. But in the case of molecular cytogenetics, it uses specialized techniques such as fluorescence in situ hybridization, that is FISH, and array comparative genomic hybridization, that is ACGH, to evaluate submicroscopic chromosomal regions. Both classical and molecular cytogenetic techniques are used to investigate constitutional and acquired chromosome abnormalities. The most recently, the next generation sequencing has been added to the cytogenetics labs to allow exact identification of breakpoints in the chromosome rearrangements. Initially, it was difficult to determine the diploid number of mammalian species because the chromosomes were crowded in metaphase stage. In the 1950s, several technical improvements such as the addition of colchicine to arrest the cells in metaphase and the use of hypotonic solutions to obtain better chromosome spreads were made. We will discuss the function and role of colchicine and hypotonic solutions when we discuss the PBLC technique in coming classes. Next, what tissues are appropriate for chromosomal study? We can use chromosome preparations for cytogenetic analysis from any dividing cells, either directly from tissue samples, for example, bone marrow, testis, chorionic villi, neoplastic tissues, or you can use cell culture for the preparation. For example, biopsy of skin or almost any other living tissues including amniotic fluid cells. But tissues preserved in a fixate tube is not suitable for making conventional chromosome preparations. This is all about the overview of the branch cytogenetics. In the next class, we will discuss one of the widely used cytogenetic technique named peripheral blood lymphocyte culture, that is PBLC, in detail. Thank you.